Hello, my name is Raha Daskabe, and I'm an instructor at Johns Hopkins Medical Institute. And today I'll be giving you a quick introduction to a piece of software we created called MEAnalyzer, which is designed to analyze spike trains from multi-electrode arrays. These multi-electrode arrays are designed to record extracellular voltages from nearby cells. They can be used for organotypic cultures or cultured cells or organoids, or even for cells outside the brain like cardiac cells or retinal cells or even intestinal tissue. They can measure acute or long-term responses under controlled conditions and are overall pretty wonderful and give you a huge amount of data. But then the problem is how to analyze it. Writing your own code is not accessible to everyone, and if you're a wet lab scientist, you shouldn't be limited in terms of the experiments you do just because you can't write code. And even if you are a coder, then you know that encapsulating software into functions is one of the central tenets of reproducibility. So here we've created a piece of software with a graphical user interface that allows everyone to easily and reproducibly analyze and visualize spike train data. The visualization part is especially important because it allows you to make observations to guide analysis of current experiments, design future experiments, and question your assumptions for things like analysis parameters. So before we go through the software, we'll go through some basic explanations. The first being that the measurements made by these devices are extracellular voltages and not action potentials. So just like any other experimental paradigm, you need to know what assumptions you're making so you can know what your potential sources of error are. MEAnalyzer analyzes spike trains, not voltages. So what is a spike and why do we discard the voltage data after extracting the spikes? Some of it is based on what's actually informative data, but there's also a feasibility issue here. So here is an example of just 60 seconds of recording at 20,000 hertz from one electrode. Now imagine that you're doing longer recordings from multiple electrodes, like 60 in this example. It's a lot of data. It's a lot of data to hold in storage. It's a lot of data to hold in memory. So what we're going to do is extract the data that we think is important. In this case, we're going to detect spikes, which are these really fast increases in voltages past a certain threshold that we're interpreting as neuronal activity. We're going to discard the voltage data and just keep the spike timestamps. And now we can process them and visualize them and analyze them. And the first thing that we're going to do is identify bursts, which are clusters of spikes. We define them, we control how they're calculated, and we can visualize them as given by the red line on top. So this is just one electrode. If we stack them on top of each other, we get a raster plot. This raster plot gives us a way of visualizing electrode activity over time over all electrodes. We can make some observations from it, but it's still not very easy to read. The good thing is that we have all the data and we can analyze it and represent it any way we want. But still, every step of the way, we need to be very clear about our assumptions. So here we can sum the electrodes in 0.2 second bins and get an idea of the spike activity over time for the whole plate. In making this binned plot, I've made an assumption about the temporal resolution I think is important. If I change that and I have 1 second bins instead, this graph will look completely different. It doesn't mean that one of these is right and one is wrong, it just means that the design is completely fluid based on my assumptions. And based on our ability to make observations, I think that this activity on this plate looks periodic. So we can move on to analyzing that. So I'm going to walk through analyzing this data with MEAnalyzer. I'm going to show the standard 8x8 plate, but there's a variety of available layouts as well as the option to just load spike trains for basic analysis and ignore spatial topography. So upon loading, the software makes some initial calculations and also plots the raster plot. This is a guide to allow you to set up the experiment. You can crop the start and end times and also select the electrodes that you want to display. The figures can be exported to whatever size you want for publication quality figures, so they don't need to be low resolution screenshots. You can also change the bin size and quickly visualize what the difference in the resulting output is when you change these parameters. Another thing you can change is what's actually plotted. So the default is to plot spike frequency in the bin times, but it can also be the percentage of electrodes spiking or the percentage of electrodes bursting. We can also look at the table of summary statistics for each electrode. The spike rate and burst rate are the ones mostly used, but I've included some other metrics that may be useful. So for example, the percent of spikes and bursts tells you about spike organization. Are they mostly random or are they organized into patterns? In some experimental conditions, you might not see a difference, so it won't be useful to you. But if you're designing an experiment interested in organization and development, this could be a useful measure. Now, some of the most ancient forms of MEA analysis are these inner spike interval histograms. I don't think they actually give you that much information. I think people are just used to them because they were an easy figure to make. I think we have much better analysis capabilities now, but I did include them because I think it's important for comparison purposes to be able to do the same analysis in a repeatable manner. So we have the inner spike interval histograms, as well as the inner burst interval histograms, and we can also look at the distribution of the number of spikes in each burst, as well as the burst length. And we can use these visualizations also to guide our parameter selection for analysis. Now, in all of these analyses, there's some information that we're missing. It's the fact that there's a spatial orientation and topography for all of these electrodes. So I've also included a method that allows us to see changes in spike activity over time, along with the locations of the electrodes. This allows us to make more observations about the data, things we might want to investigate further. What we're taking from this is that the activity on this plate is fairly synchronous, as well as periodic. So the periodicity is something we want to investigate, and I've included 2 methods for doing this. The first method is an autocorrelation, which is basically a sliding dot product with itself on the binned activity that allows you to find the main periodic interval. The second method, borrowed from the weather forecasting world, which needs to identify multiple periodic interval, is a Welch's periodogram. And in the software, you can calculate these values based on network bursts, or binned spikes, or binned bursts, and they all have different assumptions. There's also some initial methods for calculating a single peak. Another thing we can observe from the visualizations is that there's a lot of synchronous activity. We want to see if when one electrode has activity, another one does too. Graph theory is a field of mathematics that we can use to analyze networks. At the very basic level, it consists of nodes and edges, and they're combined to create a network graph. So based on the assumptions that we make, we can apply it to MEA experiments. We're first going to assume that each electrode is a node, and if any 2 electrodes are functionally connected, meaning that they have a high probability of being activated together, we connect them with an edge. Currently, this is calculated based on a cross correlation, but we're interested in implementing other methods as well, such as Granger causality. When you make these calculations, there will be a value for an edge between each pair of nodes. Here, we've thresholded them at 0.5. If we change this threshold, it changes the number of edges that are included. Additionally, each node and each edge has mathematical properties. For example, node degree tells you how connected it is to other electrodes. I've included that as a visualization, so you can change the node size and color to match node degree. The edge also has colors, and these properties are adjustable as well. You can also adjust the color schemes to a variety of layouts. Jet was the default, but we are interested in changing that because it's actually really awful in terms of visual perceptions of distances. So there it is, a quick summary of the options available to analyze spike trains in MEAnalyzer. It's available on GitHub as well as the MathWorks file exchange. Please give us a try and feel free to give us some feedback. We're currently working on implementing methods for multi-well experiments, but are open to including additional metrics based on feedback from the community.